In this video, we're going to discuss an example involving tumor sequencing. So as we talked about last time, if someone has cancer, sometimes you can sequence the DNA of a cell in a tumor, and by identifying the mutations, possibly identify a drug candidate for that patient. So in this patient, let's say that there's a mutated ERBB2 gene. Now, as it turns out, this gene is in 20 to 25% of breast cancers, this gene is mutated. And this particular gene, when it's mutated, can cause overexpression of a protein. And in this case, that's the HER2 protein. And HER2 is human epidermal growth factor receptor. So it's a receptor for a growth factor, which means that it sits on the cell membrane and it catches molecules, basically, and those molecules tell the cell what to do. Now, overexpression, just as a little reminder, that means too much. So if we have an overexpressed HER2 protein, that means we have too much of it, which in this case would mean we have too many receptors on the cell. Now, the HER2 protein causes cancer when it's overexpressed, as we just said. And this goes back to that gas pedal analogy that we used in a previous video. So ERBB2 is a proto-oncogene. So it's naturally a gas pedal. It's that part of the cell cycle that's saying, let's go forward, let's move around the checkpoint. And when it gets out of control, it's like you're flooring it on the freeway. You're going, all of a sudden you start going too fast, out of control, and as we'll see in a minute, that's how you can end up with cancer. In this case, breast cancer. So how exactly would this go down? Well, first, a patient would present with a tumor. And then perhaps it would be identified as, if it is malignant, maybe it's identified as breast cancer. And then, after sequencing, then we discover that we have the ERBB2 mutation. Since we have that mutation, we can use special drugs to attack the cancer. And in this case, that drug is called Trastuzumab, which is also known as Herceptin. And this particular drug is really good for this scenario because what it does is it blocks action of HER2. So for HER2 on a breast cancer cell, and it's only on the breast cancer cells, only the cells that are cancerous. So maybe this is our cell wall, right? That's our cell. And here is the HER2 protein. And so it's a receptor. And normally, there's a molecule out here. So normally have this molecule. It's a growth factor. And what would usually happen is this growth factor would bind to HER2, and then that would cause something to go on in the cell, probably related to the cell cycle, and cause increased cell replication and division, and ultimately the tumor would proliferate. However, trastuzumab, or Herceptin, is an antibody. And so what that's going to do in this scenario is come in and bind instead. So we don't get the growth factor. Instead, we get this antibody. So first, it blocks the growth factor. And in addition to blocking growth, it also tags the cell because we have this antibody, right, which is involved with the immune system, and when it binds to the cell, it's basically a a flag, and it tells the immune system, hey, come over here. And the immune system comes over, sees the cancer cell, realizes that it's a bad cell, and um, eventually that leads to apoptosis, which is programmed cell death, which is it's a good thing. So we have programmed cell death. And what's really cool about trastuzumab is that only cancer cells die. So this is a really promising treatment, something that we call a targeted therapy, because it only attacks the cancer cells, leaves the other cells alone, whereas with many other chemotherapies um, or radiation treatments, they often affect all the cells in that local area, which can be very harmful to the body. So this is fantastic. We've discovered a new way to approach cancer, and we've got these great treatments coming out of it, right? Well, it's not quite that easy. It's a little bit more complicated. Um, so let's talk about some of those complications. Number one, this whole thing banks on identifying mutations, but it's impossible to identify all mutations in a tumor um, or in a cell. So going back to the last video, to identify the mutations, we sequence a normal cell, and then we sequence a cancer cell, and we compare the genomes and see where they differ. And based on those mutations, then we say, oh, maybe this particular mistake in the genome is what's causing the cancer. But it's not, we're just not, we're unable to identify all mutations right now. And even if we did, we don't necessarily know what they all mean. Second, we might not have a drug. And if we don't, we don't have a drug for every case like the trastuzumab case with ERBB2. Um, so that's kind of a problem if we don't have a treatment to work against a particular cancer mechanism. And finally, there's something called tumor heterogeneity. And hetero means different. Whoops. Right, hetero means different. So basically what this is saying is that tumors are not homogeneous. They are not the same throughout. And if we look at a tumor, we have this clump of cells. But if we look closely, it turns out that these cells aren't exactly the same. They're just a little bit different. And that can be a big challenge if we're actually trying to treat cancer. And the reason that is because maybe we have this cell right here, okay, and drug X is going to work on that cell. But maybe on this cell over here, drug Y works over here. And if we only sequence the DNA in one cell, maybe the Y, the one where the Y drug works, and then we use this drug Y, we're going to kill a bunch of cells, but we're not going to kill the cells where only drug X works. So this is tumor heterogeneity, where we have slightly different cells throughout the tumor. And right now, it's a really big issue because we simply cannot sequence the DNA in every single cell in a tumor. There are thousands and thousands of cells. Now, that said, it is getting a lot more inexpensive to sequence DNA. As you might have heard in the news recently, we're approaching the $1,000 genome. There's a new machine that was unveiled that is supposed to be able to sequence a genome for only $1,000. But again, a tumor has thousands of cells, and we're not at the point where we can sequence thousands of genomes just to identify small, tiny differences. Technologically, it's not even possible, and cost-wise, it certainly is not nearly efficient. Now, that said, it's still 1 in 10 to the 9th base pairs that is incorrect when we copy a cell. And so I guess we should go back. The reason that all of these cells are slightly different is when they replicate. So as we talked about previously, when cells replicate, they can make mistakes when they copy their genome. And those mistakes can accumulate, and ultimately we get mutations. And so this is the same error rate that we were talking about with mismatch repair. If you're not familiar with that, then you should watch the video that came earlier in the cancer set. Um, but as it turns out in cancer, the rate is actually higher. And that's because in cancer, things are already a little bit messed up with the cell cycle and other cellular mechanisms. And so that can actually cause the error rate to be higher, which means that the cells are going to mutate faster and become different faster than they normally would in healthy tissues. So if we were to look at how this actually happens, start with a cell, and the cell divides, splits off into two daughters, and these daughters are actually different. Now this blue cell, this one divides, and again, one identical and then one slightly different genome. And this is just going to keep going throughout the life, and all of a sudden, all of these cells have different genomes, slightly different genomes. Now, they're not all going to be completely different, and sometimes it won't even matter, because as we talked about before, only 2% of the genome is coding, so maybe all the proteins in these cells work the same way. But it's also possible that maybe down in this cell right here, there's some proteins that are deformed, and that causes cellular mechanisms not to work properly, and therefore maybe this cell responds to different drugs than this cell would. And so this gets back to one of the big problems with cancer, which is cancer can come back sometimes. So sometimes when doctors treat cancers, it seems like the tumor goes away completely and the patient is healthy, and then maybe a year or two later, which happened with my grandfather actually, the cancer comes back. And the reason for that can be that when you treat the cancer, you get rid of a lot of cells. Maybe you get rid of all of the cells in here except for the green ones. So we missed all of these green ones. And there's only a couple of cells left, but these are resistant to the drug that you used. And now they come back. And all of a sudden now your tumor looks mostly green. And there'll be some other mutations in there, but it's mostly green cells. And now it's mostly resistant to the drug that you were using, which can be a very, very big challenge.